Click for IB Biology. We're looking at the genetic code. We're going to be looking at what are known as triplets in DNA, codons in messenger RNA, and anti-codons in tRNA, and how ultimately this all comes together to form polypeptide chains, the proteins. There's a blank version of this presentation, the PDF down in the description below. There's a 10-question quiz with answers. Channel link has got an AI prompt that allows you to initiate an individualized multiple choice assessment for yourself. Copy and paste that into a chatbot of your choice. So we've already looked at the way in which the short section of DNA which codes for a protein called a gene, how that is transcribed into messenger RNA. What we then need to think about is how the amino acids have been activated and transported by tRNA to the ribosome, where with messenger RNA, the polypeptide chain can begin to be produced. We've already learned in our protein structure that the number of amino acids and the order of the amino acids is known as the primary structure of the protein. So indeed, a gene codes for the primary structure of the protein, and translation is the process which explains how this is actually built. When we think about what the amino acid sequence might be for a particular gene, we have our gene here, and then we have our messenger RNA here. And you'll notice that I've split them into sets of 3 bases, edited in this manner, like so. And these sets of 3 bases in messenger RNA are called codons, or codons. Now, using this table here, which is a table of messenger RNA codons, we can predict what the amino acid will be for a particular sequence. And you can see that I've done that already here. So let me demonstrate how to read these tables. So for the first codon, we have AGA. The first letter is A. The second letter is G, which takes us to this box here. And then our third letter is A again, which means AGA. And you'll see that that's associated with the letters ARG. ARG stands for the amino acid arginine. Now, this table does not need to be memorized. You would be provided that in an examination. The next codon is GGG. So we come across to G here, G here. So that's identifying this box. And then here's our third letter. Let's come across, and we have glycine. So there we now know that in our polypeptide chain, we begin with arginine, then followed by glycine. CGA, C is here, G is there, A is there, and as you can see, here's CGA here, arginine again. That's surprising, I think. And we'll come to that point in a few moments. And then finally, if you would like to go through this, go to G, go to C. So we're in this box here. And then our final base is U, which is the first one. GCU corresponds to the amino acid alanine. So our polypeptide sequence is arginine, glycine, arginine, alanine. Let's look at some of the details of this. Remember that what we're dealing with are 4 bases. Now this works for codons, or it works for the triplets of DNA. And they are in sets of 3. So 4 to the power 3 gives us 64 different possible codons or triplets. And yet we only have 20 amino acids. And so we've got more code than we need for the actual structure that we're coding for. This is known as a degenerative code. And there are consequences to having such a degenerative code. We'll deal with that in just a moment. Notice also within this table that we have down here AUG is associated with the amino acid methionine. Now it turns out that all messenger RNA sequences begin with AUG, and the first amino acid brought into position is indeed methionine. However, the methionine might actually be edited away from the final protein, and we can cover that in more detail later. So all of the messenger RNA sequences begin with AUG. Then look up here, we've got 1, 2, 3 codons which are giving us a stop command. These are our terminators, and effectively give us punctuation in the translation process, indicating to the ribosome when it has come to the end of the gene copy, the messenger RNA sequence. And so with multiple codons coding for the same amino acid with punctuation, and then the start codon down here, this takes up the 64 codons which are in RNA, messenger RNA. And finally, let's consider, before we close, that the genetic code is universal. All living things use this code, and therefore we can claim that all living things have a last universally common ancestor, LUCA, and that the DNA as a coding system, since it's identical in all species, has been very, very successful and has been inherited and retained in all species. There is no other coding system. So that's one aspect to be reminded of. When we look at the coding correspondence, the 64 triplets or codons with the 20 amino acids, we've spotted the degeneracy, or sometimes known as redundancy, within the genetic system. So here we have arginine with 4 possible codons. And the interesting thing about this is that if we had, for instance, in a species CGU, a mutation which resulted in CGC would not change the amino acid, and that would buffer the mutation and protects the species. It's known as a silent mutation, or it's part of what's called neutral mutation theory. So it's a kind of protective thing. Some species tend to show a preference for some of these codons over other ones. This is known as codon usage bias. And then, as previously mentioned, we mentioned the punctuation, including the methionine with AUG in the codon. So that's our genetic code. We're now going to take a look at the process of translation itself. There's the link to the translation model, the link to the channel, which you can subscribe should you wish to, and the playlist videos are on the right-hand side there.